A trial result hits the world as a headline. A drug met its endpoint. A trial missed. A treatment showed benefit. A safety signal appeared. Investors react. Patients search for meaning. Journalists compress the finding into a few paragraphs. Clinicians ask whether it should change practice. AI systems scrape the registry, summarize the abstract, and place the result beside 100 others. But the headline is not the trial, and more importantly, the headline is not the trial record. That distinction sounds technical, but it is one of the most important distinctions in modern biomedical evidence. A clinical trial is an event in the world. Patients enrolled, sites opened, measurements were taken, amendments were filed, adverse events were recorded, data were cleaned, analyses were run, and decisions were made about what to report. The trial record is the institutional memory of that event. It is the chain of documents and data that lets someone later ask, what was planned, what changed, what was actually done, what was measured, what was disclosed, and what can legitimately be concluded. A reported result may be true and still incomplete. A registry entry may be useful and still not be the whole story. A published paper may be careful and still leave important questions unanswered. The point is not that clinical trial results are generally fraudulent or intentionally deceptive. The point is that evidence depends on the record behind the result. The first layer is the protocol. A protocol is the plan, who is being studied, what intervention is being tested, what outcomes matter, when measurements happen, how the analysis will be done, and what counts as success. The protocol records the intended design. A trial registry exposes selected structured elements of that intent to the public, while the trial as conducted records what actually happened. Those two should be connected, but they are not automatically identical. Trials change for legitimate reasons. Recruitment may move slower than expected. A safety issue may require modification. Eligibility criteria may need clarification. An endpoint may sometimes be clarified or changed for legitimate reasons, but the evidentiary meaning depends on when the change occurred, whether relevant outcome data had been examined, why the change was made, and whether it was transparently documented. None of that is automatically corruption. In real research, a clean plan often meets a complicated world. The evidentiary question is whether the record lets a later reviewer see the difference between legitimate amendment and invisible drift. When did the change happen? Who made it? Was it justified? Was it before or after the relevant data were visible? Was it disclosed in the registry, the paper, the regulatory file, or only in an internal history that the public never sees? This matters most around endpoints, because endpoints define what success means. A primary endpoint is the main outcome the trial is designed to test. Secondary endpoints may provide additional evidence, context, or supporting signals. Exploratory endpoints and subgroup findings can be useful, but they do not carry the same confirmatory weight unless the trial was designed to support that interpretation. So when a trial announcement says the study succeeded, the next question is, succeeded on what? The original primary endpoint? A secondary endpoint? A subgroup? A post hoc analysis? A composite outcome? A biomarker? A time point added later? Those differences are not academic hair-splitting, they change the evidentiary meaning of the result. A finding that was pre-specified before the trial began is different from a finding discovered after looking through the data. A subgroup result may point toward a real biological pattern, but it may also be fragile if the study was not powered to test that subgroup. A favorable secondary endpoint can be encouraging, but it should not be quietly promoted into the role of the original primary endpoint. This is why outcome switching and selective reporting matter. The lesson is not that every change proves misconduct. The lesson is that planned outcomes and reported outcomes must remain comparable. If a publication or announcement emphasizes a result that was not the original target, the record should make that visible so readers can assign the right evidentiary weight. Another distinction is statistical significance versus clinical significance. Statistical significance asks how compatible the observed data are with a specified statistical model and null hypothesis, using a chosen threshold. It does not by itself tell us whether the effect is large, durable, safe, or important to patients. Clinical significance asks whether the size, durability, safety profile, and practical meaning of the effect matter to patients and clinicians. A result can be statistically significant and still be modest in real-world clinical value. A result can miss a statistical threshold and still contain information worth studying. Neither category should be collapsed into the other. This is where headlines often flatten the evidence. Met endpoint can sound like clinically transformative. Missed endpoint can sound like worthless. The record has to do more than mark a binary win or loss. It has to preserve the endpoint hierarchy, effect size, confidence, safety context, missing data, and analysis plan that tell people what the result actually supports. Safety is another separate layer. An efficacy signal does not erase adverse events. A tolerable safety profile in one population, trial duration, or dosing context does not automatically generalize to every future use. Adverse event reporting is not a footnote to the success story, it is part of the evidentiary object. Patients and clinicians need to know not only whether something worked, but what risks appeared, how they were classified, how adverse events differed between treatment groups, how severe they were, whether investigators considered them treatment-related, and what remains uncertain. The same separation applies to registration, publication, and regulation. A trial registration makes information discoverable and structured. It can identify the sponsor, population, design, outcomes, status, and sometimes results. That is essential evidence infrastructure. But a registry entry is still a representation. It does not automatically contain every deviation, monitoring decision, data quality concern, site-level issue, or interpretive judgment that shaped the trial. A publication is also not the whole trial. It is a reporting artifact, often constrained by journal format, author choices, available tables, and editorial emphasis. Reporting guidelines exist because how a trial is reported affects how it can be interpreted. But a well-structured paper still has to be read against the protocol, registry, amendments, outcome hierarchies, safety data, and any later regulatory interpretation. Regulatory interpretation is another layer again. A regulator may weigh evidence differently from a company press release, a journal abstract, an investor note, or a patient forum. That does not mean one layer is automatically honest and the others are automatically misleading. It means they answer different questions under different obligations. The public mistake is to treat any one layer as if it were the entire evidentiary chain. Missing information is especially dangerous because it invites false certainty. Missing results are not negative results. They are also not neutral. Missingness is an evidentiary condition. Depending on the trial, it may reflect reporting delay, non-compliance, exemption, unresolved data collection, administrative failure, termination, jurisdictional limits, or a publication trail that has not been connected to the registry. The right conclusion depends on context. If an AI system scans a registry and sees no posted results, it should not automatically infer failure. If a human reviewer sees a published favorable endpoint but cannot locate the pre-specified analysis plan, they should not automatically infer deception. Both should identify the evidentiary gap and ask what the record can and cannot support. That is why this matters beyond clinical trial specialists. For patients, the record is part of informed hope. A headline can offer possibility, but the record tells what is actually known and what remains uncertain. For clinicians, the record helps separate a promising signal from practice-changing evidence. It shows whether the result fits the population, endpoint, safety profile, and comparison that matter for care. For biotechnology companies, the record is trust infrastructure. A strong result becomes stronger when the path from protocol to disclosure is clear. A weak or incomplete record can damage credibility, even when the underlying science deserves continued work. For investors, the record is a guardrail against headline volatility. A trial announcement may move a market, but the record determines whether the movement is attached to durable evidence or to interpretation risk. For regulators, the record is the substrate for judgment. They are not only reading the result, they are evaluating the path that produced the result. For journalists and the public, the record is what prevents biomedical news from becoming a scoreboard of wins and losses. The better question is not simply, did it work? The better question is, what does this evidence support and what would we need to know before saying more? And for artificial intelligence systems, the stakes are growing. Machine-readable registries and structured clinical trial data are valuable because they make search, comparison, and summarization possible. But machine readability is not the same as trustworthy provenance. An AI system can parse a registry field perfectly and still inherit a misleading evidentiary posture if the available record omits, obscures, or fails to connect amendment history, outcome changes, missing results, or safety context. If you want analysis, subscribe to Quantum Convergence Bio. architecture underneath them, subscribe to quantum convergence. The goal here is not cynicism, it is disciplined interpretation. The trial record is not bureaucracy sitting beside science, it is how science remains inspectable after the event has passed. A trial headline can tell us that something happened. A publication can tell us what investigators chose to report. A registry can tell us what was entered into a structured system. A regulatory document can tell us how an authority interpreted the evidence. But the evidentiary object is the chain connecting all of them: intent, amendment, enrollment, endpoint, analysis, safety, disclosure, and interpretation. When that chain is complete, dated, and auditable, it strengthens trust. When it is partial, late, inconsistent, or overread, it can make fragile evidence look more settled than it is. The future of biomedical evidence will not be secured only by better algorithms, better press releases, or even better individual trials. It will depend on whether the record preserves the path from plan to result, from result to interpretation, and from interpretation to decision. Because the question is not just what the trial headline says, the question is whether the record lets us know what the trial actually supports.